You heard the introduction. I don't need to repeat the topic and all. Here is the QR code where you can scan and get more into my research. I'm gonna today talk to you about how I'm using metabolomics from human, especially from larger population studies, and what I'm investigating. So I'm investigating metabolic health, and let me tell you, show you quickly what metabolic health is. Metabolic health consists of cluster of actually 5 disease condition. It's not a disease actually, but it's a condition where obesity, low HDL, hypertension, insulin resistance, high triglyceride, these are the 5 factors that we look into to take care of us as early as possible. But an individual is defined as a metabolically unhealthy if any 3 of these conditions comes together. And it's easy to understand, easy to get grasp on, and if we understand this better, we can prevent ourselves from getting into the bigger trouble of strokes, cancer, diabetes, depression, etc. So early detection of any chronic condition is where metabolic health plays a role. Having said that, I am a biostatistician. I work with varieties of data, starting from genomics, which is a large-scale, large dimensional data, till microorganisms, microbiomes, and all that. And intermediately, you can see transcriptomics, epigenomics, as we heard in the previous talk, proteomics plays also a role. I'm going to share more of insights from metabolomics and how I connect that with genomics and how I find the biomarkers or envision better understanding of metabolic health. To do so, I am specifically focusing on obesity and diabetes out of all the other conditions. Hypertension, we work with all of these 5 factors, but obesity and diabetes are my main expertise and Helmholtz research interest as well. I'm going to now show you some of the examples of my latest research. Starting with, as I said, these are the layer of omics. This is the blueprint which I use to do analysis. I gathered phenotypes, be it clinical data, clinical information, diet, food, exercise, environmental factor, etc. To combine these two, I write statistical analysis plan. Not only advanced statistics, but machine learning techniques has been used by my group. And to do by implementing this, we are in search of early markers, clusters of subjects, predictive modeling, machine learning, AI not so far so much, but we are getting into that direction. And these outputs, these findings leads us to basically 5P topic that we are into. Where the data is coming from, the data is coming from German cohort studies, several. One of them is CORA, which I have extensively used. I'm also part of pan-European cohorts. This is industry-funded projects where I am specifically working on diabetes. Not only this, I also support other big consortiums like Diamond and Diagram and Magic, which are led in Cambridge and Oxford. I'm also part of a national-wide health cohort now, where we have recruited around 200,000 subjects. And being a statistician, I have privilege to work in all range of age categories. Now I'm going to go show you some examples of what we have done recently and how interesting biostatistician's life is when multi-omics is given to us. For instance, we had a cohort where around 1,200 infants were, for them, from their serum, metabolomics were measured. And we were interested in what is the difference between formula-fed metabolome versus breastfed. What you can see here very clearly that the mothers, those who breastfed in their babies, in their infants' metabolome, more amino acids were there, were present. And in infants were formula-fed, more long-chain carbon fatty acids and acetyl carnitines were present. This is another example which brings me closer to our collaboration, not in Germany, but in India as well, where we had data from school-going kids from the adolescent age plus 7-year follow-up. Fantastically, we did the mid-lipid profiling, and what we found here is that over the course of time, the child develops obesity, and what we found is the LPCs were altered heavily. So these are the early markers that holds important to keep it in mind even in the early stage. Everything that I'm presenting today like this is already published. Another very interesting project and close collaboration with India, it was gestational diabetes cohort. Here we had women who developed diabetes during the course of pregnancy. So given a group of ladies without GDM and with GDM, we did metabolic profiling. Again, I concatenated clinical information with the different varieties of metabolites to find out that a panel of metabolites, not just one, sometimes we take ratios of metabolites, sometimes we look for the single metabolites, but in this study we find particularly panel of 8 metabolites, they were predictive of early GDM. And this is in press at the moment and will be released soon. So this is kind of gives you an overview of how I work, how my latest research is. But if you go, let me give you one example from our IMI-direct projects where I would like to show you how exactly we come to such conclusions that I have shown so far. So IMI-direct, here our objective is to look for early metabolic markers, especially in pre-diabetics and diabetes. As you can see, the whole Europe is covered. It's a pan-European consortium. Around 3,000 subjects were recruited, and for which we have the data. I mean, always the recruitment is more than what we get in the end, but in the end, we had 3,000 participants, and we were more than 40 statisticians and researchers from all over Europe come together to do this study. And I was focusing on metabolomics. So the idea here was that if we measure the HbA1c of these participants over the course of long duration, we would like to learn from the progression of HbA1c, which is glycemic deterioration, how metabolite changes and which metabolites can be as early as possible we could find which plays roles in this deterioration. This is also deeply phenotyped consortiums where we have a lot of molecular information, clinical and environmental information. So I sit down and make plan. So my first thing is I would like to see how metabolites are associated with the glycemic deterioration, right? First thing first. And as we have a time series measurement of HbA1c, we know the progression as well. So considering that, first thing first, I do the association. Not only this, beyond that, I would like to look into the mediation and the causality, like what metabolites can be mediating the factor or we are causal. And a little bit, if I get time, I'll talk about the polypharmacy effect. So to understand the association, I had for 3,000 subjects, 2 types of metabolites. One from Biocritis, which is a targeted approach, and from Metabolon I had around 800 metabolites, which are untargeted. The data was cleaned and log transformed, and as many as clinical variable I want, I had on hand, which went into the analysis. On the other hand, 3,000 patients were stratified into normals, pre-diabetic and diabetic using American Diabetic Association diagnostic criteria. And what you see here is the summary of everything. So we associated metabolites with glycemic trait. 4 tracks here shows you that we find metabolites associated with pre-diabetic, diabetic. I also threw into how pre-diabetic turns into diabetic and the progression rate. And what you can see here is something which is known. For instance, glucose. Glucose does play a critical role here in all these stages. So something that we know, if we find that in our finding, strengthens our finding and keeps us on the right track. Along with that, I found lyso-PC, what you already heard in adolescent study, playing role here in all of these 4 search. Now, if I take into a consideration non-targeted metabolomics, what I find here is massive amount of information. From unknowns till knowns, let's focus on knowns. What we find here is varieties of N-lectyl amino acids. So these are the derivatized metabolites, along with pyruvates and picolinoglycine. So pyruvate and lactate is known to be playing role in diabetes. And here, again, we are on a right track, finding right information. Along with that, we find the new information. If you look into the biochemical pathways, generally TCA cycle, which is our energy making mechanism, suffers. And because of that, alternative mechanism opens. Such as pyruvate and lactate, which was known and and was reported in our study as well. What new we found was N-lectyl amino acids, making the picture broader. Moving towards mediation and causal analysis, what we see here is we had glycemic deterioration information. We took advantage of it. And what we found here is the metabolites that we found were those who were associated were not only simply associated or correlated with the HbA1c, but they were also found in the mediating effect, like lyso-PCs and glucose. And again, from the non-targeted metabolomics, our N-lectyl amino acids, lactates, and unknowns like X something was found to be playing role in mediation. So these metabolites, the story evolves, right? I mean, they were not barely association, move towards the mediation. Now we are moving towards the causality. I wanted to know if the change in metabolites are causing change in the disease or other way around. So to do that, I have employed or used Mendelian randomization. It's a method well known, well established, which people use to understand the causality. So first question is that for me to understand if change in diabetes is causing the change in metabolites. For that, I took all the genetic variants. I dig into all the Jiva studies available for diabetes and selected my variants. Using that, I found out that from the targeted metabolites, glucose, leucine, PC are the metabolites which are causal for diabetic. And from the non-targeted metabolomics, I found glutamates and fatty acids and unknown marker. Okay, this is not very interesting. What we would like to know is if change in metabolite is causing change in diabetic, because that's where the biomarker stands. Again, the same procedure applied as before, but now my exposure is metabolites and outcome is diabetic. And of course, all the genetic variants that are interested, I considered those as instruments. And here you can see from the targeted metabolomics, I found polycholines and sphingomyelins and lyso-PCs playing role in the development of type 2 diabetes. So they are causal. And also from the untargeted metabolomics, these strange names are nothing but amino acids, omega-3 and omega-6 fatty acid, which are also proved to be known biomarker for predicting diabetes or for the causal of the diabetes. So these are the causal mechanisms that we investigated and found and published. Talking beyond, as I said, it was a PAN consortium. It was a big group. So independently, a group from University of Copenhagen associated 20 drugs with metabolomics using their multi-omics variational encoder technique. I was also part of this study, and what we found here a very mosaic and very strange patterns of metabolites given same type of drug. basically two statins specifically, which are lipid-lowering medication, gave us alternative metabolic biomarkers, not biomarkers, phenomena. So if you look into the metabolic picture, it was totally different given the same lipid-lowering medicines, which brings us closer to the point that there are certain subjects for which statins will work different, and it's worth considering. So take-home message is that globally we not only found novel metabolites from untargeted metabolomics, but also we replicated what was known. Drug association with metabolomics reveals polypharmacy, which is really, really important and upcoming field to understand how individual reacts. And I have shown you an example of like how two approach of considering targeted and untargeted metabolomics can provide us a novel marker. This work is also published and well read in the Diabetology journal. It's a no one-man show, so a lot of people are involved. I would like to thank them, thank the funders.